Scientists estimate that there are about 30,000 genes in the human genome, and unfortunately we don't really know which of these 30,000 genes are responsible for diaphragm development. So it's a challenge, and so that's what we use a broad range of interdependent genetic approaches. So we're using a bunch of different things to try to figure out how how CDH occurs. So what we do first is this is just sort of a list. We perform pathway sequencing. We do copy number analysis, genome-wide SNP analysis. We also obtain information from animal models, from hotspot chromosome regions, and from CDH-associated syndromes. And I'll describe a bunch of these in a little bit, to look for our candidate genes, or genes that we think are important for diaphragm development. So once we find our particular candidate genes, we then go ahead and we run functional and cellular assays within our CDH study group, or what we call a cohort. So I'll describe a few of these different studies that we do and how these different techniques obtain data for us that we then go ahead and assess. So as I mentioned, animal models are used to study CDH to find candidate genes. And mice are actually considered good animal models because of their close resemblance, both physiologically and genetically, to humans, and also the ease in which we can manipulate their genomes and analyze the data. So this is actually here. So this is a mouse diaphragm, but actually what I'll talk about, there's different models that we have that create CDH in mice. So I think there's about 5 or 6 right now. Different genes that when you manipulate them, they cause CDH. And this is actually one gene called FOG2 that one of our collaborators discovered, Dr. Kate Ackerman. So she found that a mouse deficient in the particular gene FOG2 results in diaphragmatic hernia in this particular strain of mice. So this is her FOG2 mouse diaphragm here. And this is considered a wild type, is how we refer to a normal mouse diaphragm. So right there, and you can see the outer, as Dr. Longoni mentioned, the outer edges, this is muscle, and this is the non-muscular region here. And you can see this is lacking proper muscularization. And this is actually where the hernia will take place, on the outer region. And also, FOG2 mice have eventrations, and that's what Dr. Longoni is talking about, that really thin diaphragm where the abdominal contents can still push through, but they just have a really thin layer over the top. So it still causes the same issues, it's just they have a really thin layer. So that's actually what this mouse has. They also have small and abnormally shaped lungs compared to the wild type, or the normal. The FOG2s are smaller, they're called hypoplastic or small lungs, and they also have abnormal lobation in the lungs. So what Dr. Ackerman did is she then went ahead and said, okay, I have this mouse model that causes CDH, so now I'm going to go ahead and we're going to sequence that particular gene in the CDH study cohort. So we did that, and she found a patient who had a mutation in their FOG2 gene, and actually the patient had an eventration defect, and the patient also had very small lungs, just like the mouse model. So that sort of clued us in on FOG2 as being important in animals as well as in humans. So now what we're doing, other investigators went ahead and sequenced the gene in there's different CDH groups, and they found more mutations in FOG2, and now we're looking at its functional role. So how does FOG2 actually, what's going on? What is FOG2 really doing during diaphragm development? So that's something that we're doing now, is to keep on looking and trying to figure out what's its actual role. So another method that we use that Dr. Longoni mentioned is called array-based comparative genomic hybridization. And we run, within our study, we run a CGH, or we call it just CGH, on all of our kids who have CDH. We just automatically run that. So we're doing basically a chromosome scan across the entire, across all the chromosomes to look for deletions or duplications, like he mentioned, of the different regions of the chromosomes. And we perform this pretty typically, as long as we have the appropriate type of DNA from the patients, or sample type, pretty much. So what we do is we take our patient DNA in the green tube there, and we add a special fluorescent probe that attaches to the DNA, and then we add a special fluorescent probe to the reference DNA. And reference is considered a patient or a sample collection of individuals who do not have CDH. So we run just the patient against a reference sample, or control is what we call it also. So we combine both of those, and we actually apply them to an array, which sort of looks like a computer chip. And the array actually has different dots, and that represents all the genome. So we're doing a whole genome scan. And then we put it into a special scanner that allows us to look at the differences in fluorescence. And when we see a yellow dot, as you can see on the scan, this means that there's no variation from the standard, meaning that the patient DNA is actually the same as the reference. So when the red and the green are combined, they fluoresce yellow. So that means that at that region, there are no gains or losses of genetic materials, no deletions or duplications. Then, if you see a green spot at a particular location, that means the patient has a duplication. Or when Dr. Longoni showed the stick-looking chromosome, you saw 2 yellows. So you have a double of that copy. You could have tons of copies of it. Sometimes people could have, 50 copies. It just depends on where you are, and that would be considered a duplication. So you have extra patient DNA compared to the reference. So the green is overshadowing the red. They both look kind of yellow up there, but and then if you have red, that means there's a deletion at that particular spot on the genome or on the chromosome, and there is reference DNA that bound, but the patient DNA is less compared to the reference. So you're missing your patient DNA there, and that's considered to be a deletion. So this is data output that we get from running a CDH scan, and you can see this is a chromosome lying flat on its side, and then this is actually the output we get, and this is the deleted region here. So if we zoom in on it, and this is actually taken from one of our patients who has a deletion on a particular chromosome. You can see that there